favorite of examiner, the gram staining. In your colleges, you must have done also the gram staining and all. If not done, remember this particular methyl gas. Okay. Sequence is important because they keep on asking the sequence. Methyl gas. And what is there in the methyl gas, guys? Methyl violet. Okay. What is there? Methyl violet. And this can be what? It can be crystal violet or it can be gentian violet. Means it should be a basic dye. It should be a basic dye like methyl violet, crystal violet or gentian violet. That you should know. G is what? G is grams iodine. Okay. G is what? That is grams iodine. A is what? A is acetone. And S is what? S is safranin. Okay. So this is a sequence which you should know. This is the sequence which you should know, that methyl violet, crystal violet, gentian violet. Okay. Either of these particular violet dyes. Then you use grams iodine. Yeah, Janvi, you can. Then A is acetone, S is safranin. If you remember this sequence, then you get up that this is the way you gram stain. The most important staining in the case of microbiology is gram staining. Okay. Okay. White color is not properly visible. White color कहाँ है? नहीं, white color कहाँ से हो गया? Okay. Okay. Sorry. It have changed to white. Fine. I will remove this color. I will remove this color from here only. Again, I'm writing up. Okay. Don't worry. Again, I'm writing up. Now I'm seeing in the chat that it is visible to me, but not to you. Okay. Fine. Do tell me, is it okay? This one is fine. Sequence is this. Is it fine now? Now it is there. Okay, fine. Methyl gas. And what do you have in the methyl gas? This is methyl violet. Okay. Or crystal violet or gentian violet. Any of the violet dye. Then you have G. G is grams iodine. A is acetone and S is safranin. Okay, S is safranin. So guys, sequence they ask in the exam and you should able to tell up that first of all we use methyl gas, then grams iodine, then acetone and then safranin. Is it fine? By staining up in this particular way, the gram positive bacteria, they look like purple to violet. Okay. And the gram negative, they look like which color? They look like pink. Okay. They look like pink. Now important is why so? Why the gram positive looks like purple to violet and why the gram negative looks like pink in color? What is the reason? If somebody asks you. This is what is the question, guys. The most important factor is thickness of the PG. What is PG? PG is peptidoglycan. Okay, what is PG? Peptidoglycan. And this particular peptidoglycan is present in what? It is present in cell wall. It is present in cell wall. Okay, it is present in cell wall. And can you tell me that which is having a thick peptidoglycan? Which is having a thick peptidoglycan? Thick peptidoglycan is a feature of gram positive and the thin peptidoglycan is a feature of gram negative. Okay. Fine. So thick peptidoglycan is a feature of gram positive and thin peptidoglycan is a feature of gram negative. First of all, remember this. Fine. So this is itself a question which you should know that, okay, this itself you should know thick peptidoglycan is a feature of gram positive, thin peptidoglycan layer is a feature of gram negative, first of all. Then I like to tell up the question which have come recently in your exam, and what is that? Teichoic acid. Teichoic acid is a feature of gram positive cell wall or gram negative cell wall. Tell me this. Teichoic acid is a feature of gram positive or gram negative. Answer to this particular question is what? Gram positive. Okay. Answer to this question is what? Gram positive. And outer membrane. If somebody asks you, the outer membrane is a feature of gram negative. This was asked in the NEET PG also and in your FMG exam also, that teichoic acid is a feature of gram positive and outer membrane is a feature of gram negative. Now how to remember? So slight bit Hindi also to manage up. Teichoic acid. Teichoic means this is a, like, how to remember this? Gram positive they have. Positive persons, they persist in life. Means in Hindi you call it टिकते हैं. They persist in life. And those who are negative in life, they get out of the life race. Remember like this. The positive persons, they stay, they persist in the life race. And those who are negative, they get out of the life race. Okay. So remember this particular one, that in the life, never lose hope of anything. If you lose hope, you lose everything. And if you have hope, you have everything. Fine. So remember this, that you have to persist. टिकना है life में positively. And outer membrane is a feature of gram negative. Is it fine? So another question. LPS, lipopolysaccharide. LPS is a feature of what? LPS is a feature of gram negative. LPS is a feature of gram negative cell wall. What is LPS? LPS is lipopolysaccharide. Okay, lipopolysaccharide. Fine. So lipopolysaccharide, that is there in this particular one, in the gram negative cell wall. And remember this particular one for the lifetime, that because of this only, what is being produced by the gram negative ones? Endotoxin. Okay. Because of this only, what they produce of, guys? Endotoxin. And that's why the endotoxin stuff leads to what? Endotoxic shock. So if somebody asks you that which particular bacteria are there which sends you in the endotoxic shock, the answer is gram negative. Okay, they are negative people. They send you in the shock. Is it fine? So endotoxic shock is mostly seen in infections of gram negative ones. E. coli, Klebsiella, Salmonella, all them, they leads to you to endotoxic shock and all. First of all, remember this. And from which particular thing? There is a antigen which is there, which was asked in the NEET PG also, can be asked to you also. Somatic antigen and flagellar antigen. Two particular names. Somatic antigen and flagellar antigen. The somatic antigen is termed as O and flagellar is termed as H. Somatic antigen is termed as O and flagellar is termed as what, guys? H. Fine. So this particular somatic antigen, it is being derived from lipid A of lipopolysaccharide. It is being derived from what? It is being derived from lipid A of the lipopolysaccharide, and this is the flagellar antigen. It is donated by, like, which particular one denotes this? It is being denoted by this particular H. Actually, these are Greek words, one and houch. O is somatic antigen, flagellar antigen is H. Fine. So this question can be asked that this O antigen is derived from where? Lipid A. And this flagellar antigen terms what? H. Flagella. Those particular ones which have flagella, that is this particular flagellar antigen. Is it fine? So remember this particular fact also, that somatic antigen they derive from lipidae, and flagellar antigen is denoted by which particular alphabet? H. Yes, very right. In the Vidal test, we use the antibodies for them. We use antibodies for them. So guys, what we have remembered methyl gas. This is to be remembered. Gram positive purple, gram negative pink. Most important factor, thickness of peptidoglycan layer. Number of other factors are also there. No time to discuss them. Remember this, that the most important one is thickness. Okay. Then I have told you positive persons, they persist, टिकते हैं, outer हो जाते हैं कौन? Life race से. Negative people. Lipopolysaccharide is a feature of gram negative cell wall, and this LPS is the one which leads to this endotoxin production, which later on leads to what? Endotoxic shock. Somatic antigen is there, flagellar antigen is there, which is going to be talked about in the future. So this is being derived from lipidae, and this is H. This is H. Fine. So this much we have done. Now, the second stain, if you want to know the important one, the second stain is acid fast stain. Acid fast staining or acid fast stain. This is the second one, acid fast staining. And acid fast staining, you know that you do for what? You do for Mycobacterium TB, or those who have which particular acid in them, guys? Mycolic acid. Those who have what? Mycolic acid or Mycobacterium TB mainly. We do the acid fast staining. So in this, what is the sequence? The sequence is, first of all, you have the sputum smear. Okay, first of all, what you have, guys? Sputum smear. Then the carbal fusion. Fine. Then you use up for the TB 20% H2SO4. Okay. And then you have methylene or methylene blue. Okay. Methylene or methylene blue. So this is the sequence which you need to know regarding the acid fast staining, carbal fusion, and after the carbal fusion, what you do, guys? Intermittent heating. What you do? Intermittent heating. IH. Intermittent heating. Now you can ask up, sir, why we do intermittent heating? We do intermittent heating because they have mycolic acid. Okay. Until you remove this mycolic acid, the dye will not come inside. So that's why you need intermittent heating. Okay. Again, the sequence. How to remember the sequence? Car. Inside. Hai. Which color? Blue. So this gives you a sequence. Car. Inside say. Hai. Which color? Blue. Fine. Because what they will do in the exam, they keep on jumbling up. The carbal fusion, then they will put methylene blue, then we'll put H2SO4. Like this, they keep on jumbling this. So you should know that first of all, you use a carbal fusion, then you do intermittent heating, then you use this decolorizer. Which one? 20% H2SO4, and then you use what? Methylene or methylene blue. And when you do this, what you observe, guys? You observe a particular field in which the background is what? Blue, and against that background, pinkish to reddish color, acid fast bacilli. Okay, pinkish to reddish color, acid fast bacilli you observe, means in the exam, how to identify that you're dealing with acid fast staining, gram staining and all that. Acid fast staining hack is what? You have to see the background. If the background is blue, against that you are seeing pinkish to reddish thing, anything, that is what is what? That is Zn staining. Okay, that is Zn staining or acid fast staining. Is it fine? So if you are confused of that, what is Zn? Zn staining is the other name of acid fast staining, and Zn is the name of two scientists, Zeal and Nielsen. Okay, so remember this particular fact, guys. Car inside means from inside. Hai, which color? Blue. This gives you a sequence. Okay, and another important which nobody tells, actually they don't know, means in which particular power you see this. Gram staining and Zn staining, in which particular power you observe? You observe in the oil immersion lens. Oil immersion lens, and that particular one is 100x. Shom Shudra had told perfectly right, 100x. So that is the one in which you observe gram staining and Zn staining. And this question might come. It is having the maximum magnification because 100 into 10. 10 is of eyepiece. Okay. And 100 is of this objective lens. So 100 into 10 is 1000. 1000x is the magnification of this oil immersion lens. Is it fine? So remember this particular fact that acid fast staining is an important staining which you should know. The sequence you should know, and you should know that how to identify. Identify by seeing up the background. Background is blue, against which you see what? Pinkish to reddish color, acid fast pass line. Is it fine? Now, another important fact which you should know here is that there is a cold method of staining also. There is a cold method of staining also, and this cold method of staining is quinone staining. Okay, quinone staining. Peace Wealth, the other name of acid fast is ZN, Zeal and Nilsson. Okay, Zeal and Nilsson, these are the two scientists who have modified the acid fast staining. Okay, who have modified the acid fast staining. That is also termed as ZN staining. Is it fine? So cold method of staining is modification. Modification of what? ZN, Zeal and Nilsson. And this is cold. Here I told you na, that you have to heat up, intermittent heating. But here K for cold you have to remember. That quinone staining we do not heat. We just increase the concentration of carbonyl fuchsin. We increase the concentration of carbonyl fuchsin. Is it fine? So two important staining we have talked about. One is Gram and one is this. That is the acid fast staining. Is it fine, guys? Am I okay with this? Fine. The third most important one which you should know is regarding the Albert. Okay, Albert staining. Albert staining. Albert staining you do for what, guys? Albert staining is being done for Corynebacterium diphtheria. That is being done for what? Corynebacterium diphtheria. And this particular one stains it green. Okay, it stains with which color? Green. Along with that, it stains the Babe's earnest granules. The Babe's earnest granules also, I will show you the pictures also of this, don't worry, later on. Okay, initially I'm just telling up that which all things are important and all, and later on we'll go by the image wise, and that will help you to rebuild that thing also. So Babe's earnest granules. So remember like this, guys, that Albert is a person. Okay, Albert is a person who have gone where? To China. Why I'm telling that fellow have gone to China? Because Corynebacterium diphtheria is having which pattern? That is Chinese letter pattern. It is having a Chinese letter pattern. Okay, it is having a Chinese letter pattern. So Albert is a guy who have gone to China. Okay, and there that fellow have eaten up corns. Okay, there that fellow have eaten up corns by putting up which particular color chutney? Green chutney. Okay, so he have made a great effort to have gone to China to eat up what? Corns. By putting up which chutney? Green chutney. Why I have told you this? Because this particular one comes in the exam and they try to ask the color. Okay, so now you will remember that the Corynebacterium diphtheria stains of which color? Green. By which particular staining? Albert. And this particular Albert have gone to which country? China. Is it fine? And this particular one, like VG will cover up most of the microbiology topics. Don't worry. Okay. So this particular one, that is the Babe's earnest granules, which are also termed as polymetaphosphate granules. Okay, which are also termed as polymetaphosphate granules. Okay, they are stained up bluish black. Okay, they are stained up bluish black or brownish black. Fine. So Albert staining, I have told you. I have told you acid fast staining. I have told you